Okay guys, we're ready to start. For those of you that are watching from somewhere else, I'm going to take some time and introduce myself and tell you a little bit about my lab. We do have some students from Blue Ridge Elementary School who are watching us live, and they will be able to ask questions during the presentation. So my name is Jessica Merricks. I am a PhD student here at the University of Missouri, and I study acoustic communication in frogs. Okay, so our lab is really interested in understanding how frogs communicate. Now you may wonder why anyone would actually care about why frogs communicate. Well, there are a couple of reasons why frogs are really excellent for understanding communication. Communication is everywhere. Animals communicate for lots of different reasons. We have to communicate for mating and for communicating with family members. Communication is just all over the place. And frogs are a really great model for studying communication. If you think about different animals, so think about different animals that can communicate. You have things like crickets, insects, small animals that communicate. Then you have things like frogs. And then if you get really complicated, you can think about how humans communicate. Well, frogs are kind of in the middle when we think about complexity. So insects, crickets, and grasshoppers, their communication is pretty simple. And humans, our communication is obviously very, very complicated. So frogs fall right in the middle of that. So scientists are really interested in understanding how communication works in general. And so by using a model like frogs, we can get a better picture of how communication really works. So our lab is interested in the evolution of communication in frogs. Frogs mainly use communication for two reasons. One, males will communicate to other males to indicate some sort of aggression. So males may make sounds to other males if a male is in their territory. That's one way that they communicate. The most important reason for frog communication is actually for mating. So males produce a species-specific call. Every species has its own unique call. It sounds different from any other species. And females are actually able to listen very, very carefully and recognize the calls of their own species. This is actually really kind of amazing. What I want you to think about is imagine you're in a room. My 4th and 5th graders are in a room right now with a bunch of other people. Imagine that you and one other person in that room speak English, and everybody else, let's say there's 100 people in the room, everyone else is speaking a different language. And your job is to find the one other person in the room who speaks English. That's about what it's like to be a female frog in a breeding pond. So a female makes her way to a pond. It's really extremely, extremely loud, and she has to find the one other individual that speaks her language, so to speak. So these frogs are really very skilled at doing this. And as a scientist, we want to know how that works. So to do our research, there's actually two parts to our research. First, we go out into the field. And what I mean by that is we actually go outside where the frogs live. We go out and we find the frogs, we collect them, we will record them and take measurements in the field. And then we actually collect some of those frogs and bring them back to our laboratory, which is where I am right now. This is my laboratory. We can ask very specific questions about how frogs communicate within the lab. So what you're going to see now is actually a trip that I took with my lab out to a local pond. It's called Ashland. The pond is in the city of Ashland. It's called Basket Wildlife Area. And that's actually where we went out and collected some frogs just a few days ago. Okay, so the next thing you'll see will be me at one of my field sites. You'll get to see exactly what it looks like, and you'll get an introduction to the frog that we're going to be studying today. Okay, so I'll see you back here in just a few minutes. We are here at the Basket Wildlife Area. This is the pond where my lab comes to collect frogs. So hopefully you can hear in the background the frogs are already calling. Right now the males are making their way to the pond, and the females who are still out in the forest, they're foraging, looking for food and trying to stay moist. they are going to make their way to the pond as the sun starts to set. That's where the males and females will start to pair up. So some species call it a really high frequency, really high pitch, and some call it a really low frequency. Some species have a call that's made up of lots of different little pulses, and some species don't. There are a lot of differences between the different species, and we want to know what in particular do females listen to. So are females listening to the frequency? Do they care if the sound is high or low? Or are they more interested in how the call is timed? Are there lots of pulses, or is it just one continuous sound? So we come out here and we collect the calls of the males, and we also collect females. And once we go back to the lab, I'll show you how we figure out exactly what the females are listening to. This is a male spring peeper. You can tell it's a male because he has a dark throat sac. So his throat sac, his vocal sac is down here. He was just calling, so it's a little bit loose. Females don't call at all, so they don't have dark throat sacs. And also females are going to be a little bit larger than this. So it's really easy to tell males from females. The tympanum is located right behind the eye, and that's actually where his eardrum is. So they don't have external ears. Their eardrum is directly behind that layer of skin, and that's called the tympanum. Right now, the chorus is just really, really loud. There's tons of males out, and they're all calling, trying to attract females. Okay guys, I hope you enjoyed that very quick trip to the pond. Now I have the females that we collected here in this big refrigerator. It looks like a big refrigerator. It's actually called an incubator. Okay, it's an incubator. And this actually is set to a very specific temperature to keep our frog's body temperature right where we need it to be. So I'm not going to leave it open very long, but I do want to show you what it looks like in here. Okay, so I have individual boxes. And there's an individual frog in each of these boxes. Okay, we're actually going to test 3 frogs today. Alright, I'm going to take one out just really quickly so you have a chance to see. Alright, this is a female spring peeper, just like the one you saw in the video. She might jump, so I'm not going to leave this out too long. Okay, I do want to ask you guys a question though. Hopefully my 4th and 5th graders remember this. Why is it that temperature is so important for frogs? So think about, this incubator is a little bit colder than it is outside. Can someone think about what that might do to the body temperature of the frog? Take a minute and think about it. Have one of your teachers call on you and come up to the microphone, and I'll see what you have to say about that. Okay?
Body organs, and makes them wait till it gets warmer to where the sugar comes out of their organs. And
very good, Gable. So Gable remembered what I told you about how the body temperature of the frog changes with the temperature outside. Okay, so the blood of the frog actually changes in response to the changing temperatures. So at really, really cold temperatures, the frog's body temperature gets really, really cold, and some frogs can actually freeze, like they can freeze solid. I'm really excited that you remembered that. Very good. So I have the frogs in here. They're set at a certain temperature, and they stay that way until we get ready to run our experiment. Now, I do want to say one more thing about the females. The females that we have are gravid. that word just means that the females are actually full of eggs, okay? So our females are ready to mate at this point. When we collected them from the field, they were actually in the mating position with a male, okay? So these females are very much ready to get rid of these eggs. We have to keep them in this incubator and keep them at a certain temperature so that those females don't drop their eggs. Basically what happens is when a female shows up to a pond, she's full with eggs and she's ready to mate. If for whatever reason she doesn't get a mate, she will actually just drop her eggs. She'll just waste them. And so there won't be any babies in this case. So it would be very wasteful for the female to release her eggs. So we have to keep her at a certain temperature so that she doesn't waste her eggs. Now, this would be bad for us if she did waste her eggs because females are not interested in our experiment if they're not full of eggs. So we want to make sure that they stay full of eggs so that they respond in our experiment. Also, we don't really want to waste the frog's eggs. Her job is to go to the pond and reproduce and make more frogs. So we don't want to be responsible for our frogs dropping their eggs. That's why we have to keep them at a certain temperature. So now I'm going to show you what I think is the coolest part of my lab. I'm going to bring you right over here. Okay. This big metal box here is called an anechoic chamber. That's a really big word, but all it means is it's a soundproof chamber. All right? So I'm going to take you in here and show you the features of this chamber and why we use it for our experiments. Okay, so we're going to go in here. It's a little bit dark. You may not be able to see perfectly well, but I just want to give you an idea of what it's like in here. Okay? So this is inside the anechoic chamber. And if you could be in here with me, you would notice that you actually cannot hear your own echo in here. So there's foam up in the ceiling, there's foam all along the walls, and that prevents sound from bouncing off of walls. Okay, I can try to move that so you can you guys see the foam? It's like little triangles popping off of the wall. All of that foam is set up to keep the sound from bouncing off the walls. So when we put the female in here, we're going to play sounds to her to see what it is that she really likes. She will be hearing just a perfectly pure sound, no echoes, no sound bouncing off walls. So the way this works is when we get ready to run our experiment, we take the female and we put her down here on the floor. So there's a white sheet down here, okay, and there's a cup. So we actually put the female in this cup and we cover her up. This is sort of heavy so she can't get out. And what happens is there's a speaker on this side of the chamber and there's another speaker on that side of the chamber. So what's going to happen is I'm going to put the female inside this cup. I'm going to leave the room and turn the lights all the way off. She won't be able to see a single thing, okay? All that she will get is the sound that we're going to play to her. And so what we really want to do is we want to ask the female a question. Which sound do you really like more? Do you like that high sound or that low sound? Okay, so when we run the experiment, I'm not going to be in here, but we do have a camera in here. So if you look back here, you might be able to see that infrared camera. So that camera basically can see in complete darkness. So even though she can't see anything, we will be able to see her. Okay, so let's go back out and I'm going to show you a few more things about the species and our research question, and then we'll actually run the experiment. Okay, this is a picture of the spring peeper. Can you guys see the, are you guys able to see the PowerPoint right now? Okay, great, sorry. Alright, so, okay, this is a picture of the spring peeper. I'm going to play you a sound so you can know exactly what it sounds like. You heard it at the pond, but I'm going to play it again for you. Okay, so that is a typical spring peeper male call. So this is what we're going to be working with. Now, I mentioned at the pond that frogs differ for a couple of different reasons. First, they differ in the timing, okay? So what you're looking at is a picture of the spring peeper call. So everywhere where you see these green lines, that's actually the sound that you just heard. Okay, so I'm going to play it again. I want you to watch this graph. This graph is actually called an oscillogram. It's just a picture of sound. So you should be able to hear it and tell where there's sound and where there's silence. Oops. Here we go. Okay, so you can tell the timing of this spring peeper call. He makes a little peep every couple of seconds. I'm going to contrast this with a different species just so you can get an idea of how different the frogs can really be. This is a river frog. Okay, so he's a lot larger and he sounds completely different. So I'm going to play a call of the river frog and here's the picture of it. So try to follow along with your eyes as the call is being played. Alright. I'll play it one more time since it's really low. Okay, so that's what a river frog sounds like. Now you can tell there are obviously very important differences between the spring peeper and the river frog calls. Now, in addition to differences in the timing of the call, we can think about the difference in the frequency. When I say frequency, I'm just talking about the pitch. Okay, so you can think about notes on a piano. The higher you go up the piano, the higher the frequency or pitch. And the lower you go on a piano, the lower the frequency or pitch. Okay, so what you're looking at now is a different type of graph. This is called a spectrogram, and it shows exactly where the frog is putting all of its acoustic energy. What I mean by that is every time the frog makes a sound, it's using energy at different frequencies. So this is a picture of a spring peeper call, and if you look at the really bright orangish yellowish spots, that is exactly where all of the energy is for that particular call. So spring peepers have a frequency of more than 2,000 hertz. Okay, that's the value that we use to measure frequency. So they're at right about 2,000. Now let's look at the river frog. Okay, so this is the same thing. This is a spectrogram. The bright orange, yellowish color shows where most of that frog's energy is. And you can see for this graph, all of the energy is way at the bottom, right? So this call is much lower frequency. So you can think of the spring peeper as like being a high note on a piano, and the river frog being a low note on the piano. Okay, so there are timing differences and frequency differences. Now I want to take just a few minutes and talk about how frogs actually process sound. Okay, we have external ears, right? You can see the ears of a human. I tried to show you the ear of the frog when I was at the pond. I'm not sure if you were able to see it, but they don't have external ears. They have something called a tympanum, and that's basically just an eardrum right on the side of their head. So what you're looking at now is actually a cross-section of the ear of a frog. So if you see here where it says airborne sound, that white structure there, that's the tympanum. So everything from there onward to the left is inside the frog. All the way over to the left, that's where the frog's brain actually is. Okay? So the sound travels through the tympanum. It goes all the way back to the frog's eardrums. Frogs are really cool because they actually have 2 eardrums. Humans only have 1 eardrum. The 2 eardrums, they have 2 different names. One of them is called the amphibian papilla, and the other one is called the basilar papilla. So I just put up a red box to show you exactly where I'm talking about, and we're going to zoom in on the next slide so you can see it a little bit closer. Okay, so we're zoomed in. Here is the amphibian papilla, so that's one of the eardrums. And here's the basilar papilla. There's the other eardrum. The amphibian papilla is actually responsible for processing low frequencies. right? So like that river frog has really low pitches. The amphibian papilla is actually responsible for processing those low sounds. The bacillar papilla is responsible for processing high frequencies, so those high pitches like the spring peeper call. So this is really interesting, and scientists are really interested in understanding how these two eardrums work. We know that different frogs have different pitches, and we know that females are really interested in whatever it is that their species is doing. So if their species is calling really high, they're going to be more interested in that than they would be in a low sound. At least that's what we think, at least. So here's our research question for today. Since spring peepers have two eardrums for hearing different frequencies or pitches, if we add extra frequencies to the call, will the frogs like it more? So scientists are interested in knowing if both of those eardrums are being stimulated at the same time, if females would actually find that call more attractive than a regular call, which would just be stimulating one eardrum. So you guys are going to help me answer that question today. I'm going to just do a quick demonstration to show you how the experiment will work. You don't need to write anything down right now. I just want to show you what's going to happen. So this is a bird's eye view of the chamber. This is where I was just standing a few moments ago. You'll notice on the left there's a speaker, and then there's another speaker over on the right. That black circle in the middle is just indicating where we would be putting the frog for each experiment. And there's a little camera at the bottom just to show you that that's where we're going to be watching from. So we'll put the frog in the chamber, close the door, turn off all the lights, and we will play sounds from both of these speakers. So she'll hear a sound from the left side, and she'll hear a sound from the right side. And what's going to happen is I'm actually going to pull a string outside of the chamber, and that's going to raise up that cover on her cup. Once she is free to move, she's going to listen to those sounds and decide which one she likes the most. Okay, so what happens is when the cover gets lifted off the frog, we actually are going to time how long it takes the female to get to whichever speaker she chooses. So let's say we put the frog in the center. You guys will get to watch what happens. Let's say she does this behavior. Okay, so let's say she makes her way over to that right speaker. What we would write down in our data notebook is, 1, that she chose the right speaker, and 2, how long it took her to get to that speaker. So for trial number 1, we would say she chose the right speaker, and we'd write down how long it took. Let's try another one. For trial 2, the female does this. Okay, so in this case, the female chose the left speaker. So in our data notebook, we would write down that the female chose the left speaker, and we would write down how long it took her to get there. There's one other possibility that might happen, and I want you guys to be prepared for this because this is a live experiment and these are live animals, and they will pretty much do whatever it is that they want. So here's another possibility of something that might happen. Yeah, so in this case, the female actually just completely ran away from the arena. She left the arena. We call this a no choice. That just means that the female is actually just trying to get away. She's more interested in getting away from the situation than she is in actually mating. So in this case, we would write down that the female did not make a choice, and this behavior would not count towards our data analysis. So that data point would essentially be thrown away. Okay, so I just want to show you a picture of the stimuli that we're going to be using today. Again, this is just an oscillogram. Again, I'm just showing you what I will be looking at, and you will be hearing this, but you won't be able to see this picture. So this top line where you see that big bar and then there's a smaller one, that's the sound that's going to be coming out of the left speaker. The sound down here, this singular bar, that's the sound coming out of the right speaker. So the female will be hearing the left speaker and the right speaker alternatively and continuously until she makes a sound, or until she makes a choice. Alright, here's what the stimulus will sound like. Once you get to the video, you'll hear this exact sound. Okay, and so that sound will be repeated over and over again until our female actually makes a choice. So that's one test that we'll run. Here's the other test. You'll notice it looks a little bit different. This is actually testing a low frequency stimulus. Okay, so you have this long bar and then a short one. That's the left speaker. And this is the right speaker. Okay, so I'll let you know what that sounds like so you'll be aware when it comes. Okay, so you might notice that it sounds a little bit different. The first stimulus, I'll go back to it. This is actually a spring peeper call. So this shape is the actual normal spring peeper call. And this is actually a high frequency sound that we have added to the call. Okay, so regular spring peeper call plus a high frequency sound. That's coming out of the left speaker. On the right speaker, it's just a regular spring peeper call. Nothing fancy, just a regular spring peeper call. Again, for the second stimulus, we have a regular spring peeper call, and then we're actually going to play a low frequency sound. Okay, so regular peeper call, low frequency sound, that comes out of the left speaker. And then in the right speaker, it's just a normal spring peeper call. So we really want to know which of these two choices the female is going to like more. Okay, so now that I've gone through and I've explained the experiment, I'm going to give you guys a chance to ask me any questions you might have. And while we're doing that, I want to make sure that everyone is ready to collect their data. So I'm not sure if we passed out papers or if the students are using their science notebook, but at this point we need to have that in front of us so we can be ready to go. If you have any questions, I'm ready to answer them. Make sure when you ask your question, you say your name and speak nice and loud for me. You don't have to press the button.
If you add a high frequency thing or a low frequency thing, why would it make a difference?
Okay, that's a very good question. So the question was, if you add a high frequency sound or a low frequency sound, why would it make a difference? So again, I want you to think about the two different eardrums that the female frog has. So we know that one of the female's eardrums is being stimulated by a regular spring peeper call, right? The spring peeper has a high frequency call, and so we know when the female hears that eardrum is being stimulated. What scientists don't know is how sensitive her other eardrum is. So we know that she doesn't really use it very much because spring peepers don't make low frequency sounds. But we are interested to know if she can actually process that sound. So even though spring peepers don't make those extra high frequency and low frequency sounds, we want to know if by having her eardrum stimulated by those sounds, if she actually finds those sounds more attractive. Are there any other questions?
Just talk right into your mic. You don't need to press the button. Tell her who you are.
Myra, and how many times are you going to test the frog?
Okay, thank you for your question, Myra. We're going to run 3 experiments today. So I showed you all of those frogs I have in the incubator. We have 8 of them. We don't have enough time to run all 8 of them. So we're actually going to run 3 different frogs today. Okay, any more questions?
Jason, how long does it take for a female frog to notice a male frog?
That's a good question, Jason. So the question was, how long does it take for a female frog to notice the male frog? Did I get that question right, Jason? Just want to make sure. Okay, so it really depends. So in a really loud chorus in nature, remember I said there's lots of different sounds going on. It's very, very loud. Sometimes it can take the female quite a bit of time to find a sound that she really likes. She's very good at being able to pick out calls that she finds attractive. So as soon as she's close enough to a frog that she is attracted to, she can get there pretty quickly. What you might see in the video, if these frogs are really excited, they may just run straight to the first sound that they really like. So sometimes it can only take a couple of seconds, and depending on the situation, sometimes it could take longer. When we run our experiment, the female has 2 minutes. So we start the sound, and she has 2 minutes to make a choice. If she doesn't make a choice in 2 minutes, we throw that data point away. We sort of assume that if she is hearing both sounds clearly and she hasn't made a choice in 2 minutes, that probably means that she's not very interested. So that's our timeline for running the experiments. Anybody else?
No. Anybody else have questions? Not at this time, Mrs. Merricks.
Okay, great. So now I'm going to show you the data table. Now you guys should have this in front of you. I'm just going to help you guys get it all set up together, okay? Okay, so you should have something that looks like this, and we're going to go through together and fill it in, and then we'll start the experiment. So today's date is April 24th. So everyone go in and write down today's date if you haven't already. And it's 2013. So the experiment that we're running is we want to know if frequency is important to female preference. So do females like high frequencies or low frequencies added to the spring peeper call? So I'm just going to call this female response to different frequencies. Okay, so I want you to write that down as well. So the name of our experiment today is female response to different frequencies. And of course, we are testing the spring peeper. So once you get done writing down the title, you have to write down what species we're working with, because there are lots of different species that we study in our lab. And in our lab notebook, we have to make sure that we are writing down the right species so that we know exactly what we're studying. So get those 3 things written down. I'll give you a minute to make sure everyone's caught up. And teachers, while the students are writing, if you could please ready the timers, or whoever is designated to run the timers for the first experiment. We're about ready to get started, and we'll need to make sure that those students are ready with their timers in hand. Now, in our data table, we have to list a few things. First, we have to list the frog ID number. Our frogs, spring peepers, are listed like this: PC, which stands for Pseudacris crucifer. That's the scientific name of spring peepers. And then we give every frog an individual number. So this is the first frog of this experiment, so this frog will be PC.01. Okay, so under frog ID, you should write PC01. And we're actually going to study 3 frogs today, so you can go ahead and write PC02 and PC03. Okay. The first test that we're going to run today is the high frequency stimulus. So under test name, you would just need to write high frequency. And you can abbreviate it so you don't have to write out the whole word. Okay, so now what we're going to do is we'll cut back to the live feed so you guys can see the box, because I'm getting ready to start the experiment. So I have the first frog. This is PC number 1. She's ready to go. So what's going to happen is I'm going to take the frog and put her in the chamber, and I'm going to cover her up, and you'll see this. I will warn you that inside the video feed inside the chamber is not the best. So you may not be able to see the frog perfectly clearly, but you should be just watching for the dark black spot moving across the screen. So if the frog moves to the right, that means she chose the right speaker. If she moves to the left, then that means she chose the left speaker. So what you're going to do, those of you that have the timers, as soon as you see the cover lift off the frog, start your timer. That means that the female has heard the sounds and she's free to make whatever choice she wants to make. So we're going to go through this together. I don't know what's going to happen, okay? So we all have to pay very close attention. And I have my timer here, so I'm going to be timing her as well. After we run her, I'll come back and we'll talk about it. Okay, so everyone get ready. Make sure you're paying close attention. Teachers, if you can make it as dark as possible in your room, that will make the video a little bit clearer. Okay, so I'm going to hop up. I'm going to go put her in. And the next thing you'll see will be the feed from inside the chamber. Okay, be right back. Alright guys, I hope you were able to see that. I would like a volunteer to come to the microphone and tell me exactly what happened. Make sure you tell me who you are.
Brayden. And the frog went to the right side.
Very good, Brayden. So the frog went to the right speaker. Okay. Now again, that right speaker was playing just a regular spring peeper call, and the left speaker was playing a spring peeper call plus a high frequency sound. So can someone tell me what time you guys got? How long did it take her?
Steven and I got 1:25.
Very good. I got 1 minute and 24 seconds, so very good. So we are actually going to switch back to our data sheet. Okay. And the female chose the right speaker. And so that was 84 seconds. So 60 seconds plus 24 more seconds is 84 seconds. So write that down in your data table. Okay, now we're going to run the next frog. This is PC number 2, and we're going to do the same test. So this is high frequency. Okay, so make sure you have written down PC number 2 and high frequency. Now I'm not going to switch back to the live video, but I have the second frog right here in my hand. So the next thing you'll see, it'll be dark, and as soon as the cover is lifted off of the frog, start your timer and watch what she does. Okay, guys, that was the second experiment. I would like a volunteer to come to the microphone and tell me what happened.
This is Dara. I think he went to the right.
Very good, Dara. So the second female, PC number 2, also went to the right speaker. Very good. Can someone please tell me how long it took this female?
It's Emily, and it took about 1 minute and 18 seconds.
That's exactly what I got, Emily. Very good. I got 1 minute and 18 seconds. So I'm going to go back to our data table so we can enter that in. So we're going to write it in seconds. So it took 60 seconds plus 18 more seconds is 78 seconds. So get that written down in your data table, and we'll be ready to move on to the next one. Okay, someone just give me a cue when you guys are ready. Okay, I need everyone to focus on the screen. The very next thing you'll see will be PC number 3 running through our last experiment. And we're actually going to run a low frequency test this time. Okay, so everyone ready your timers and keep a close look on the frog.
Not so much.
It's coming after you again.
Okay, everybody. So, someone volunteer to tell me what happened.
This is Dawson, and it went off the side.
Very good, Dawson. So we would consider this to be an escape behavior. So our frog was more excited to run away than she was to actually find a mate. So she was not attracted to either one of those calls at this very moment. So I'm going to go back to the Excel sheet. And right here under Notes, we're going to write NC, which stands for no choice. Okay, so this particular data would just be thrown away. This doesn't count at all. So with the last few minutes that we have, I want to get you started on a graph of this data. We will not get it finished during the live video, but I hope you'll have some time to finish it when we get done with the video. Okay, so the title of our graph is going to be the same as our experiment. So, female response to different frequencies. So what you can do right now is, on your chart, go ahead and write the title. Now, for this graph, we're just going to graph the time it took each female to get to the speaker. So down here on our x-axis, we're going to label this frog ID. Okay, so that this is going to be each individual frog gets her own bar. Now remember, when we do graphs, we always skip that first column. So we're going to skip this column. And we're going to label the second column PC01. Okay, you want to skip every other column. So our next one is PC02 and then PC03. Now, can I have a brave volunteer tell me what the y-axis will be? So I have a box here covering up what I have put here, but I want to know what you would put going up that y-axis.
Trent, and I say the time.
Very good, Trent. So we would put the time that it took the female to get to the speaker. So I'm going to put time to speaker. Okay, and I've already labeled mine. So I went all the way up to 100 seconds. Oh, and be sure we write the unit, right? So we measured our time in seconds. So make sure you add that as well. Okay, so again, I'm not going to go through the whole graph with you, but frog number 1, PC number 1, took 84 seconds. So we would make a bar going up to 84 seconds. So I'm going to do that quickly. And you guys can take your time and make sure you do your graph nice and neat. So there's my first bar for the first frog. It's a little bit more than 80 seconds. So once we're done with the video, we're going to have some time for questions and then we'll close. You can go ahead and finish your graph. One thing I would recommend that you do is use 2 different colors because we did 2 different tests. Okay, so PC number 1 and PC number 2, they both responded to a high frequency stimulus, and PC number 3 responded to a low frequency stimulus. So you would want to use a different color and show that those are 2 different experiments. Once you're done with your graph, I want you to write one conclusion. So I just want you to write one thing that you learned from this broadcast. Okay, so we are essentially done with the experiments. We do have some time where I can answer some more questions. If anyone has any questions about the frogs, what I do, anything at all, we can take some time and answer some questions.
Be sure you identify yourself and speak clearly. You don't have to press the button.
My name is Isaac, and I want to know what the last two frogs' names were.
Hi, Isaac. The frog names? I'm not sure I understand your question. So each frog goes by a number, right? So the first frog was PC01, right? The second frog is PC02, and the third is PC03. And we just keep going up the line. So when I get ready to test the next one, it'll be PC number 4.
My name is Caitlin. Would the female frog go to a sound that it heard before?
Oh, that's a really good question, Kaylin. So the question was, will the female go to a sound that she's never heard before? Actually, there's a lot of research going into asking that very same question. We are really interested to know if female frogs are attracted to new sounds. So what this essentially means is that in the brain of a frog, when they hear a sound that they've never heard before, their brain actually gets stimulated more sometimes. So by hearing a new sound, it might be more attractive to that female simply because it's new and she's never heard it before. There may also be the opposite effect. It might be that when she hears a brand new sound, that she really dislikes that sound and she would move away from it. So it depends on the sound and it depends on the frog, but that's something that a lot of frog researchers are really interested in figuring out. Okay, if there are any more questions, I'm ready.
My name is Mackenzie, and if the frog was the same species as another frog, and they heard a new sound, would they go to the same species of frog as they are, or a different one?
Very good question. So Mackenzie wanted to know if the frog hears a frog sound like her own species, or something else, a different frog species, which one would she go to? Well, for the most part, our research has shown us that female frogs will more than likely go to the frog that sounds most like her own species. This is really important because frogs have to be really careful to mate with the right species. So it might be really bad if a frog made a mistake and mated with a different species. It might be that their offspring don't survive, or that their offspring may not even hatch at all if two different species actually mate with each other. Now, sometimes mistakes happen and frogs mate with different species, and you end up with hybrids. Now, I don't know if you guys remember some of the research I talked to you about, but I actually study hybrids. I actually have a few of them right here. So these are a few of my hybrid frogs, right? So these are 3 females. You're just looking at her belly there. There's one in the corner, too. But these guys are a hybrid cross between 2 different species, right? So sometimes these frogs do make mistakes in nature, and 2 different species will mate together. And sometimes it works out okay. My frogs are perfectly healthy and happy. So sometimes that does happen. But again, to answer your question, for the most part, females are going to prefer the call or the sound that their species is making over anything else. Anybody else have a question?
This is Dawson, and do you ever use any male frogs and then put in female sounds?
Oh, very good question, Dawson. So Dawson was asking me if we ever do this experiment with males. So do we play sounds to males? Well, we don't play female sounds to males because females actually don't make any sound at all. But we do play other male sounds to males. So I told you that males will communicate aggression. So they actually will, sometimes they actually even fight with each other. So there are people that are interested in understanding how that works. So you could run a similar experiment. You could put a male frog in the chamber and play different aggressive sounds to that male and see what he does. One thing we've learned, which I think is really very interesting, is if you put a male in the chamber, and if this male is really quiet, he doesn't call very much, he's actually known to go towards a sound that is really high quality. So let's say you have a male out there that's a little bit wimpy, maybe he hasn't had very much to eat, his call isn't very attractive. What he will actually do is he will go and move towards another male that actually has a really nice, attractive call, and he'll just kind of sit there and wait for a female to come by. So this is actually something that does happen. Males do respond to the calls of other males. Good question. Anybody else?
It's Emily, and why do females only mate certain times?
Very good question. So Emily wanted to know, why do females only mate certain times? So that's just based on the reproductive biology of these particular frogs. It takes females between 1.5 to 2 years to actually reach maturity. So after they've metamorphed into frogs, it takes them about 1.5 to 2 years to where they're actually able to reproduce. And the timing for that is all based on hormones. So as the seasons change, the hormones in the female frog change, and she starts to develop hundreds and hundreds of eggs in her belly. And when that happens, the signal goes off in her brain that she needs to go and find a mate. So that usually happens, each species has a different breeding season. Spring peepers, they start right around the end of February, early March, and they go right around to about now, so about the end of April. So all of the frogs, all of the spring peepers either have already mated or they will be mating very, very soon, and then those hormones turn off and there's no interest in mating anymore until the next season comes around. Good question. Any more? Okay, one more question.
This is Myra, and were the sounds that the frogs were hearing, were they just recordings?
Oh, that's a good question. So Myra wanted to know about the sounds that we were just playing and if they were recordings. So what we do in our lab is those pictures I showed you of those calls, those are actually artificial. So we actually have a program on our computer where we can create the frog sounds. So what you were hearing was a computer simulation of the frog calls, not the actual recording. The reason we do that is because we want to make sure that the frogs are hearing just a pure sound, no background noise, no wind blowing or anything like that. So we use these pure sounds so that we are really, really testing the female's response to just the frog call. That's also a really good question. Okay, guys, well, it's time for us to go. I really hope you enjoyed your experience here. I'm looking forward to answering more questions when I get to school. And, yeah, I hope you guys had a great time. See you later.
Thanks, Mrs. Merricks.
You're welcome.